Hello everyone, welcome to this online lecture on blotting techniques in genetic diagnosis. Myself, Dr. ArulJothi from SRMIST of Science and Technology. This lecture will give overview on southern blotting, northern blotting, and western blotting, and how these techniques are used in genetic diagnosis. These blotting techniques play a crucial role in genetic diagnosis and also helps us to understand the genetic variations, uh, differential RNA expressions, and differential expression patterns of proteins. So we can use these techniques in a genetic diagnosis effectively, and we'll see how we are doing it. Yes, understanding the genetic mysteries. We can understand the genetic mystery at different levels of these macromolecules. For example, we can use southern blotting to understand the problems of the DNA or at the DNA level. We can use northern blotting to understand the dynamics of mRNA expression, and we can use western blotting to find the expression patterns of the protein. Altogether, these three techniques gives insights into the genetic disorders at DNA level, RNA level, and at the protein level. For those who don't know the central dogma, basically any information or any phenotype is passed in the same order that is shown in this slide. We have a chromosome, and the chromosomes have got DNA, and these DNAs will be transcribed to mRNA, and this mRNA will be translated to protein level. If there is any wrong or anything goes wrong in this process or in the information, then it leads to a genetic condition. So we can detect the genetic condition at the chromosomal level by using karyotyping or FISH or other techniques, and we can diagnose the genetic condition at the DNA level by using various techniques, and southern blotting is one such technique to detect the DNA mutations or variations at the DNA level. And coming to the RNA, so RNA is basically the amount of RNA decides the amount of RNA expressed decides the outcome of a phenotype. So we should be able to measure the quantity or the expression levels of mRNA by using northern blotting. And same applies to the protein level also. We can quantify it, and sometimes we can qualitatively tell if it is present or not by using this blotting. Coming to the southern blotting is a technique used to detect specific changes in the DNA sequences. It involves DNA fragmentation, gel electrophoresis, transferring it to the membrane, and hybridization with a labeled probe. Coming to the labeled probe, these labeled probes are prepared in specific to some DNA sequence. It can be a gene or can be a marker. So these labeled probes should be prepared beforehand. So for southern blotting, you need to prepare your sample, and you need to have this apparatus that is shown on the screen, where you'll be doing now horizontal gel transfer, where you have to run the samples in Agarose gel electrophoresis, and you have to run and separate the fragments as shown in the image. Yeah, this explains how southern blotting is done. So basically, the genomic DNA is isolated and been digested into small fragments and run through agarose electrophoresis, and the DNA in the agarose electrophoresis is being transferred to a membrane, and this membrane is taken for further analysis, where you can see we have a probe which is designed to detect specific DNA sequence. Okay, so when we add this probe onto this membrane, if the DNA sequence is present, it will go and bind to the specific site, and we can observe it in the when exposing the membrane to a X-ray sheet. We can observe the signal. For example, if the gene is not present or if there is something deleted majorly, we may not be able to identify the pattern. You can have a look at this right hand side blot. This blot is for detecting Huntington's DNA, where in the first lane you can see it is a control lane which shows the normal size of the Huntington gene. Whereas in the second lane you can see there is a normal gene present. In addition to that, there is an expanded which has got 36 repeats more than the normal one. And the third line is normal, fourth line is normal, whereas fifth line has got a bit more expansions, trinuclear expansions. So changes like this can also be captured using sudden blotting technique. The next slide is we'll talk about northern blotting. So basically, northern blotting is same as the DNA southern blotting, except the fact that it uses RNA as the X material. So same principle. It involves RNA isolation and separating it in a agarose gel and then transferring it to a membrane and hybridizing with the probe. Yeah, you can see the pictorial representation of northern blotting where you see the RNA is being separated based on the size and we are transferring the RNA into a membrane and this membrane is being hybridized with the radio labeled probes. And remember, these probes also should be specific to the gene or mRNA and which can be visualized under the Excel line. Remember, the RNA, the mutations or the deletions in the RNA may not be captured here, but the RNA can be quantified by using this. Maybe not an absolute quantification, but a relative quantification we can do. Based on the expression levels of the RNA, we can speculate the cause of the disease. You can see in this slide, you can see after doing all this northern blotting, you can see that in the first slide, in the first lane, the RNA levels are very low, and the second lane is a bit more and it's increasing and increasing. So for example, if these four samples are from different patients, we can assume that this can be a normal expression level, whereas these two patients, patient 1 and 2, they have a reduced or down-regulated gene expression for this specific gene, which may cause the specific disease. So RNA expression levels can be assessed using northern blotting. Next is Western blotting. Western blotting is a technique that is used to identify and quantify specific proteins in a sample. It involves protein separation using a polyacrylamide gel and transfer of the protein to a nitrocellulose membrane. And instead of probe, we will be using antibodies specific to the target protein, and we use the antibody conjugated with the radio-labeled probe or any fluorescent probe or even HRP conjugates to detect the protein level. This can be used as a qualitative and sometimes quantitative. Uh, yeah, we can see how the Western blotting is used in genetic diagnosis. We'll take the example of Duchenne's muscular dystrophy, where we assess various human skeletal muscle proteins. For example, lane 1, this is a control, and lane 1 and sorry, the patient 1 and 2, they are from the congenital muscular dystrophy patients. You can see that these two patients they lack laminin alpha 2 chain of merosin protein. Merosin protein is an extra cellular matrix protein which helps in the anchoring of the dystrophin that gives the muscle cell integrity. So this protein, in case of 2, the protein is completely absent, whereas in case of patient 1, the protein level is very less. Similarly, coming to the case 3 and 4, which is sarcoglycanopathies, a form of Duchenne muscular dystrophy. You can see that in 3, the sarcoglycan is completely absent, and in case of 4, it is very low. Okay, so the loss of these proteins led to sarcoglycanopathy. And in case of lane 5, compared to the control, this lane 5 is a patient of Duchenne muscular dystrophy. You can see there is no dystrophin protein and less levels of laminin and reduced levels of sarcoglycan and reduced levels of dystroglycan compared to the control. So this led to the muscular dystrophy. So in this slide, we can clearly say that Bayesian blotting technique can be used effectively in genetic diagnosis. Of course, not all the cases, but in specific cases where we have clear distinct pattern of proteins that gives idea of the genetic condition. In those cases, we can use Bayesian blotting. So coming to the application aspect, these blotting techniques are widely used in clinical diagnostics to identify genetic diseases, genetic mutations, and gene expression patterns between tissues and between different patients, and even find the protein abnormalities. And these techniques are very important in diagnosing genetic disorders, sometimes infectious diseases, and cancer. So altogether, these techniques provide valuable information for personalized medicine and to strategize the treatments. So what are the challenges and limitations that we may face using these techniques? Challenges like most of the techniques are time consuming and will have big procedures, long lengthy procedures, and sometimes we may not have enough sample for diagnosing or for running the gels, and sometimes we may have technical variations. So overcoming these challenges, we can integrate other complementary techniques to enhance the accuracy and efficiency of genetic diagnosis. For example, we can even go for a sequencing or MLPA to detect deletions, or we can learn other techniques to just uh, complement these challenges. So the future directions of genetic diagnosis lies, as I told you, integrating the advanced techniques, complementary techniques with the existing blood techniques that may enhance the sensitivity of these techniques. For example, using next generation sequencing or bioinformatics or other precision medicine approaches. This will converge so this convergence will enable comprehensive genetic profiling, personalized treatment strategies, and improved patient outcomes. Discussing about the clinical impacts of these blotting diagnostic techniques, it extends to the realm of personalized medicine, where genetic information or the information obtained about the mutations uh, through these techniques guides us for targeted therapies and use of proper treatments and disease management. For example, uh, if you have a, mild form of Duchenne muscular dystrophy or muscular dystrophy, that is not actually because of the loss of dystrophin, but other axillary proteins. You can alter your treatments based on the mutation. I don't have to go through the generalized Duchenne's muscular dystrophy treatment. So these blood techniques will give us some idea on how to take the treatment and how to use what therapies to go. So definitely give us idea on personalized treatments. Coming to the conclusion, these blotting techniques are indispensable tools in unraveling the genetic mysteries and diagnosing the inherited diseases. Their continued advancement and integration with the emerging technologies holds great promise for precision medicine and personalized healthcare. And thank you so much. If you have any questions, please write to me at this email ID. And thank you for your patience.